So, hello, my name is Enrique. I'm a third-year student, PhD student, and I will be presenting part of my research work entitled Assessment of Polyhydroxyalkanoates Production in Photosynthetic Communities from Activated Sludge and Chlorella sorokiniana consortium. Now, to begin with, in recent years, microalgae have been under the spotlight to produce bioplastic. This is due to the fact that these microorganisms, and when I say microalgae, I mean green algae and cyanobacteria, they mostly produce biodegradable polymers such as polyhydroxyalkanoates or PHAs and polysaccharides. In short, polyhydroxyalkanoates or PHAs are this type of polyesters or biodegradable polyesters that consist of hydroalkanoic acid monomers that are accumulated intracellularly and can be used to produce plastic-based products such as packaging products or disposable cutlery. In contrast, polysaccharides are these rather complex long chain molecules that consist of monosaccharides and again can be accumulated intracellularly and also within the cellular wall. And this can be used not only to produce bioplastics but also in the biofuels and in the food industries. Now, today, the most common way to produce this or to induce the production of these biodegradable polymers in microalgae is through a two-stage or filtering process, or also called feast and famine. In short, what we need to do here is that the microalgae, the inoculum, needs to be first inoculated in a nutrient-abundant medium in order to just basically produce biomass. And then after this first feast process, then we induce or re-inoculate the microalgae into a nutrient-deficient medium, most commonly nitrogen, in order to trigger a mechanism known as nitrogen chlorosis, which actually not only reduces the chlorophyll content, hence the yellow color here, but also induces first the polysaccharides production and then the polyhydroxyalkanoates production in the long run. However, one of the current technological limitations that the technology has is that this technology is kind of species dependent, meaning that certain species can only do this type of, accumulate, especially PHAs, and it's usually done with commercial strains, which are actually hard to maintain and to scale up. In view of this, different sources are being screened, and one of these are activated sludge. Activated sludge has, again, been very well searched because during this procedure, not only it has been found that we can find photosynthetic microorganisms that can actually form predominantly photoeutotrophic consortia under persistent illumination, but also during the same activated sludge procedure, this procedure is, let's say that it's prone to nutrient fluctuations, which actually put a selective pressure on the microorganisms, which in few words, is just actually promotes the survival of the most malleable, of the most resilient, adaptable, and possibly polymer accumulating microalgae. In this case, our objective of this research work was to explore the polyhydroxyalkanoates and polysaccharides production yield in mixed photoautotrophic consortia for bioplastics production under nutrient-impaired conditions. Now, in order to do so, we conveniently split our methodology in three different stages, beginning with the generation of a photosynthetic community. This was done first by sampling activated sludge from a wastewater treatment plant in Querétaro, Mexico, and being grown under the growth conditions here described. Then we also recover a freshwater creek sample, which was kindly provided by the Sao Paulo University, which came from, as I said, a eutrophicated freshwater creek, and it was also grown in nutrient medium or BG11 nutrient abundant medium in order to promote their growth. Then, after procuring this first photosynthetic consortium, we then proceed to the screening or the identification and retrieval of the polymer accumulation or the polymer storing algae. For this, all the consortia that were generated in the first step, we actually harvested or centrifuged them and rinsed them with distilled water in order to eliminate any residual nutrients. And then we subjected these consortia to a nitrogen-free BG11 medium in order to promote the accumulation of polymers. So during this step, the screening for polymer was done qualitatively, basing ourselves on the Nile red staining, which basically gives us a kind of a disfluorescence seen here under a fluorescence microscope. And in any case that any strain actually had this bright red fluorescence seen here, we will actually retrieve all of this through micro manipulation under the microscope and then transfer them to BG11 medium in order to promote their further growth. Then finally, we proceeded to enhance the polymer production of these selected consortiums by virtually doing the previous step that I mentioned before, the two culturing step, under the conditions or the growth conditions seen here. So basically, first we induce the selected consortia to a nutrient-abundant BG11 medium in order to promote their growth, and then we actually transfer them to a nutrient-deficient medium in order to promote then the polymer accumulation. During this last step, we actually quantitatively assess the polyhydroxyalkanoates and polysaccharides production through the acidic methanolysis GC method and the Dubois method, respectively. Now, as part of our results, what we observed on the first hand, I will mostly focus on the results of our last phase. So basically what we observe is that in the selected consortia, especially the one from the sludge-borne photosynthetic consortia, we observe that during the first 14 days, we observe an exponential increase in the accumulation of polyhydroxyalkanoates under these nutrient pair and CO2 enriched conditions. The same behavior was also observed for the mixed Chlorella sorokiniana consortia, which came from the freshwater creek. And in this case, well, as I said, the biggest accumulation was done or was achieved on day 14, more or less, and the production yield was 200 micrograms of PHAs per gram of dried biomass per day for the sludge-borne and 300 micrograms of PHAs per gram of dried biomass per day for the mixed Chlorella sorokiniana. Now, the same behavior was also observed for both of the consortia, now this time for the polysaccharides. In this case, we also observed that polysaccharides achieved their highest accumulation on day 14. In this case, the accumulation or the production yield in the sludge-borne photosynthetic consortium was of 13 milligrams of glucose per gram of dried biomass per day. And in the case of the mixed Chlorella sorokiniana, it was 10 milligrams of glucose per grams of dried biomass per day. Now, what really stands out about all of this chart is that basically both of these polymers actually had a really similar behavior. And now this is interesting because usually polyhydroxyalkanoates are usually seen as carbon reserves and energy storage reserves, which are used under nutrient limitation. However, with our results, we are actually seeing that polyhydroxyalkanoates may actually have other functions within the cell, such as a transitional electron sink or a structural support during cell division. All of this assumption is based on the fact that actually if polyhydroxyalkanoates and polysaccharides were both only carbon polymer storing molecules, one or the other will be enhanced or will be improved, but not both. Now, in terms of total production, we've seen that, in fact, the polysaccharides production was actually improved in comparison to previous study, especially for the freshwater-borne consortia. In this case, we observed a 21% increase in the polysaccharides production, whereas in the polyhydroxyalkanoates production, we actually observed not a rather similar concentrations or total yield. This could be mostly because of other stress factors that could be affecting the production of polyhydroxyalkanoates. By reviewing the literature, what we found is that when a constant injection of air or oxygen occurs during growth, the medium actually is under a really oxidative or is under an oxidative stress. This oxidative stress actually overwhelms the cellular defenses against the reactive oxygen species, which are actually formed during this oxygenation. And, well, basically during this type of mechanism, what occurs here is that under this excessive oxygenation, the cell actually uses most of these reducing agents and energy to reduce or to reduce the amount of reactive oxygen species instead of using all of this to produce PHAs. So as a conclusion, basically what we can say is that waste sludge is a novel source of cyanobacteria and green algae that could be used for bioplastics productions, that nutrient impairments such as nitrogen starvation and phosphorus limitation and CO2-enriched air injections allowed an increased production of biopolymers in photosynthetic consortia, and ultimately that the accumulation of polyhydroxyalkanoates and polysaccharides is not mutually exclusive and could be potentially enhanced by complementing nutrient deficient conditions with other methods, such as using methodologies that could reduce the amount of oxygen dissolved in the medium, such as profiting for microalgae bacteria associations, or by inducing other micronutrient deficiency, which actually could impair the energy requirements of the ROS production.